You know, when we think about aging, I feel like we usually picture it like a car slowly breaking down.
Right, yeah. The classic wear and tear model.
Exactly. Like the parts just get old, the engine starts to sputter, the transmission slips. We imagine it as this inevitable, almost chaotic decay where the actual physical components just stop working.
Yeah, I mean, we tend to assume the cellular blueprint just gets smudged over the decades. The machinery degrades and things fall apart simply because the materials are old.
But think about a symphony orchestra for a second. If a performance of a Beethoven symphony goes completely off the rails, it's almost never, because every single instrument on stage suddenly snapped in half.
No, the physical instruments are totally fine.
Right. The catastrophic failure usually happens because, like, the violin section stopped listening to the cellos, or half the room stopped looking at the conductor. The parts aren't broken, but the communication is just a total mess.
And that analogy, honestly, perfectly captures a massive paradigm shift happening right now in biology. We're finding out that the instruments inside ourselves aren't necessarily broken. It's the coordination between them that's failing.
Which brings us directly to the stack of research we are unpacking today for you. We have an incredibly detailed 2024 study published in Nature Aging by researchers Liotte, Lopes, and Bayer.
Yes, titled Loss of Coordination Between Basic Cellular Processes in Human Aging.
And our mission for this deep dive is to figure out what actually happens to our genes as we age. Like, do they just get tired and make random mistakes? Or is there a hidden systematic shift in how they work together?
It's a huge question. And to start, we really need to understand the old assumption.
Right, because for the longest time, scientists believed aging was driven by an increase in transcriptional noise.
Exactly. And to understand that noise, we have to look at transcription itself. It's the fundamental process where your DNA is read and copied into RNA.
Which then acts as an instruction manual.
Yeah, exactly. It tells your body how to build the proteins it needs to survive. So the noise theory basically suggested that as we age, the enzyme reading the DNA RNA polymerase just starts getting sloppy.
It reads the wrong parts at the wrong time?
Yeah.
Or it fails to turn off?
Gene expression becomes increasingly random. It's like a radio dial slowly drifting off the station and dissolving into static.
But the researchers behind this Nature Aging study, they wanted to challenge that specific idea. They didn't just want to look at single genes emitting static. They wanted to measure coordination across the whole network.
So they built a gene regulatory network model, a GRN.
And they trained this baseline model using data from 1,443 cancer cell lines, which is where I actually had to pause my reading.
Yeah.
Because if we're studying healthy human aging, training a baseline model on cancer seems entirely counterproductive.
It sounds totally backwards, I know. Cancer is famously chaotic.
Yeah, it's mutated. Why use that as a baseline?
It's a counterintuitive approach, but methodologically, it's actually quite brilliant. Think about whole tissue data. Say you take a tiny biopsy of liver or brain tissue. You are looking at a highly complex, messy mixture of dozens of different cell types. You got neurons, glial cells, blood vessel linings, immune cells, all mashed together.
So it's too noisy to see the fundamental rules.
Exactly. It's incredibly difficult to extract universal mathematical rules of how genes interact from that kind of noise. But cancer cell lines grown in a lab, they provide incredibly pure, high-volume data.
Oh, I see. So they use cancer cells because the sample is just vastly cleaner to read.
Cleaner, and crucially, it's still constrained by the laws of physics. Even a chaotic cancer cell has to obey fundamental cellular mechanics.
They can't just break the laws of biology.
Right. Specifically, it has to obey strict stoichiometric requirements.
Stoichiometric. Let's unpack that term for a second.
Sure. Think of stoichiometry like the precise recipe required to bake a cake. You need exactly 2 cups of flour for every 1 cup of sugar.
If you mess up the ratio, you don't get a cake.
Exactly. If a cell is building a vital protein complex out of gene A and gene B, it needs those proteins in an exact unchangeable ratio. If it doesn't meet that ratio, the cell dies, even a cancer cell.
Wow, okay.
So by training their model on over 1,000 of these pure cell lines, the researchers established the universal rules of how genes literally must coordinate together to keep a cell alive.
They essentially mapped the foundational rule book of genetic coordination. And once they had that rule book, they took it and applied it to healthy human data.
Yes, specifically the GTX dataset.
Which is this massive, rich database covering 948 healthy human donors. donors, aged 20 to 79, across 30 different biological tissues. And they introduced a specific metric here called predictability.
Right, using a statistical tool to measure rank correlation, specifically Spearman's row.
Let's break down Spearman's row, because that sounds intimidating. What did that actually measure?
It's just a way to measure how well the relationship between two variables can be described. It doesn't look at the raw numbers, but the rank or order.
So it's like, if variable A is the highest it's ever been, is variable B also the highest it's ever been?
Exactly. So they would look at a specific target gene in a healthy person and then look at its regulatory neighbors, the genes that the rule book says it should be working in tandem with.
The model asks a simple question, right? Based on what the neighbors are doing, can we predict what the target gene is doing?
You've got it. If the regulatory neighbors turn their expression volume up to an 8, does the target gene also go up to an 8?
And if the observed behavior perfectly matches the prediction, that gene has high predictability.
Exactly.
Going back to our orchestra, if the violin section drops their volume to a whisper to build tension, the cello should be doing the exact same thing.
Right.
If I hear the violins drop to a whisper, I can confidently predict what the cellos are doing. But if the violins get quiet and the cellos are suddenly blasting a chaotic jazz solo, predictability drops.
And that drop in predictability is exactly what the researchers were looking for as they analyzed the tissue data of individuals moving from their 20s into their 70s.
Okay, so let's do a quick recap of what we've covered so far. Aging isn't just random static or noise. Scientists can actually measure how well our genes coordinate by comparing real tissue data against a universal regulatory model.
A model based on strict biological recipes.
Right. And I want to ask you, the listener, to think about your own body for a second. Have you ever noticed certain systems seemingly falling out of sync as you or your loved ones age? Like maybe sleep cycles totally decoupling from energy levels.
It's a very common, deeply human experience.
It really is. And this study is essentially showing us the genetic blueprint of that exact feeling. But what fascinated me is that the data completely dismantled the old wear and tear theory.
How so?
Well, if the whole body was just slowly decaying into static, you would expect this predictability metric to drop universally across every single tissue.
Oh, right. But it didn't. The tissue specificity was wildly unexpected.
Break that down for us. Where did it drop?
In some tissues, coordination absolutely did drop as people aged. The blood, the thyroid gland, the tibial artery in the leg, and visceral adipose tissue.
Which is the deep fat stored around your internal organs.
Exactly. In those specific areas, the genes were clearly losing their coordination over time. The cello stopped listening to the violins.
But then in other tissues, the predictability actually increased. The coordination got noticeably tighter as the donors aged.
That was a huge shock.
Where did they see that happening?
This happened in the testes, the mammary tissue of the breast, the mucosal lining of the esophagus, and arguably the most complex organ of all, the brain.
Seeing the brain's genetic networks conform more tightly to their regulatory models as individuals age.
Yeah.
I mean, that challenges so many pervasive assumptions about cognitive decline.
What's fascinating here is that the brain is largely composed of postmitotic cells.
Postmitotic, meaning they don't divide.
Right. Neurons generally don't divide and replace themselves the way skin cells do. You kind of have what you have.
So because it has to survive for a lifetime with the cells it has, the aging brain appears to compensate by hyper-regulating its existing networks.
Likely to maintain crucial synaptic connections, yes.
Which leads us directly into a major paradox the researchers highlighted, the variance paradox.
Oh, this is a brilliant part of the paper.
The data showed that interindividual variance, meaning how different gene expression levels are from one person to another, generally increases with age across almost all tissues, except for the artery and thyroid.
Right. So you and I might have nearly identical gene expression profiles today in our 20s.
But by the time we hit 70, our gene expressions will look completely different from one another.
Exactly.
But wait, if variance between people goes up so drastically, shouldn't predictability go down? Doesn't more variance simply mean more chaos?
It is an incredibly intuitive assumption, but understanding why it's wrong is the key to this entire study. High variance does not equal biological noise.
Okay, explain that.
The researchers discovered that the specific genes displaying the highest variance between individuals actually showed increased predictability.
How does that physically work? If our genes are doing totally different things, how are they still behaving predictably?
Because the variance isn't random error. It is a highly calculated adaptation. Think about the span of 50 years.
Okay.
You and I will experience different diets, different stress levels, different sleep habits, and different environmental exposures.
Right, our bodies accumulate different types of cellular damage.
Exactly. The variance we see in the data is our genetic network shifting into entirely new states to deal with our specific environments.
Oh, I see. The environment forced the conductor of your orchestra to play a completely different song than mine.
Yes. The song is different, that is the variance, but every single instrument in your body is still perfectly coordinated with the others to play that new song.
So the variance isn't sloppy regulation. It is a customized, adaptive response. The model can still perfectly predict the target gene's behavior because all of its regulatory neighbors are shifting their expression together in a united front.
You nailed it.
Time for another quick recap. Aging affects tissues differently. Some lose genetic coordination, while others, like the brain, actually tighten it up.
And increased gene variance between older individuals is usually a coordinated adaptive response, not random error.
So, question for you listening. Knowing that your brain actually increases genetic coordination as you age, does that change how you view cognitive aging? It's pretty wild to think about.
It's very helpful, honestly.
It is. So if our bodies are doing this incredible thing where the variance between us is actually a coordinated adaptation, how does that play out when we zoom in on specific cellular departments?
Well, the study didn't just look at whole tissues. It drilled down into specific biological functions. And this is where the core mechanism of aging really reveals itself.
Right. They analyzed within module coordination versus between module coordination.
Exactly. Within module means genes inside the same cellular department working together. Between module represents different departments talking to each other.
And the major finding was that the relationships inside a functional group often hold remarkably strong. But the relationships between different cellular processes break down catastrophically.
Let's look at the specific example the researchers focused on, the mTOR signaling pathway.
We hear about mTOR constantly in longevity research. It's the mechanistic target of rapamycin. It basically serves as the master regulator of cell growth and nutrient sensing.
Right. When nutrients are abundant, mTOR sends the signal to build, multiply, and grow.
And the researchers specifically zeroed in on a gene called LAMTOR5. This gene produces a protein that's part of something called the regulator complex,
which sits on the membrane of the lysosome.
Okay, let's unpack that. How does that physical structure connect to cellular aging?
We have to look at the physical space of the cell to understand why this matters. The lysosome is essentially the cell's stomach. It's an acidic recycling vat that breaks down waste.
Okay, got the visual.
The regulator complex physically anchors to the outside of this vat and acts as an environmental sensor. It's constantly tasting the environment for amino acids.
Pacing for food.
Right. When it senses that you just ate and there are plenty of nutrients available, it activates mTOR. It essentially tells the executive suite, we have building materials, initiate growth.
But biological growth requires a massive amount of energy. So mTOR normally has to be in constant, tight coordination with the mitochondria.
The powerhouses of the cell. They produce ATP, the basic unit of cellular energy.
Right, and here's where it gets really interesting. The communication fails.
Precisely. The researchers found that in older tissues, specifically the aging tibial artery LAMTOR5, loses its regulatory correlation with vital mitochondrial genes.
We're talking about specific genes like COX14, COX6B1, and NDUFB3. What do those actually do?
Those genes are the blueprint for the electron transport chain.
Which is the actual mechanism inside the mitochondria that generates the energy, right?
Yes. The electron transport chain passes electrons down a line to pump protons across a membrane, creating a gradient. It's much like water backing up behind a dam.
And releasing that water drives the turbines.
Exactly, that gradient drives the production of ATP. Now, mTOR also has to coordinate with V-ATPases.
What are V-ATPases?
They are tiny molecular proton pumps, like ATP6V1F, that help manage the acidity required for these processes.
Okay, so normally the nutrient sensor on the lysosome, the proton pumps, and the energy generators in the mitochondria are all in lockstep.
But with age, the predictability plummets. The regulatory relationship just vanishes.
It perfectly mirrors a massive corporate office. You have the executive suite, which is mTOR, looking at the quarterly nutrient reports.
They see raw materials, so they start shouting, aggressive growth, build more infrastructure.
Exactly. But the emails they are sending down to the manufacturing floors of mitochondria are just bouncing back.
Yeah.
The executives have no idea that the factory floor is completely out of the energy required to fulfill those orders.
And the physical result of that disconnect is devastating for the tissue.
Because the cell is trying to build without any power.
Right. The cell is constantly receiving aggressive growth signals, but it lacks the energy output to properly fold proteins or clear out waste.
So damaged parts start to accumulate.
Yes, and the cell eventually enters a state of senescence. It stops functioning but refuses to die and just starts secreting inflammatory signals.
And it's fascinating that this decoupling happens so aggressively in the tibial artery. The arteries in your lower legs are under immense mechanical stress.
They have to constantly expand and contract to pump blood against gravity for decades.
That requires incredible amounts of energy. When the energy grid decouples from the maintenance signals in a high-demand environment like that, the tissue just degrades.
It is a profound point of failure. The decoupling of nutrient sensing from energy production is a hallmark of why our tissues eventually fail us.
Okay, let's recap this section. The biggest failure in aging is this between module communication, specifically the decoupling of the cell's nutrient-sensing growth pathways from its energy-producing mitochondria.
The executives lose touch with the factory floor.
Exactly. So a question for you all listening. Where else in life do you see systems fail? Not because the individual parts broke, but because the communication between them just stopped.
It's a universal vulnerability coded right into our DNA.
It really is.
Oh.
But the narrative doesn't end with this systemic failure. The body doesn't just passively accept this loss of communication, it actively goes on defense.
This is perhaps the most encouraging part of the nature aging study. While we see this terrifying drop in between module communication, the researchers documented that within module communication can simultaneously get much stronger.
I was looking at the specific subnetwork of genes in the blood that the researchers highlighted. The predictability of these genes skyrocketed with age.
Yes, the serum-specific proteins.
Right. We have ALB, which produces serum albumin. We have apolipoproteins like APOA1, APOA2, and APOB, which are responsible for lipid transport.
And we have members of the coagulation cascade, like F2, which produces thrombin, and FGA, a subunit of fibrinogen.
These are the absolute heavy hitters of blood clotting and vascular defense. But why would these specific genes lock into a much tighter, hyper-coordinated pattern as we get older?
If we connect this to the bigger picture, it comes down to a physiological phenomenon known as inflammaging.
Inflammation and aging combined.
Exactly. As human beings age, we tend to accumulate a rising tide of systemic low-grade inflammation throughout the body.
Because of those senescent cells we mentioned earlier.
Partially, yes. The immune system becomes slightly dysregulated, and senescent cells like the ones in the arteries secrete those inflammatory molecules into the bloodstream. The body is highly intelligent. It recognizes this rising baseline of stress.
And the study pointed out another crucial set of genes that tighten up alongside the clotting factors. Genes producing inter-alpha-trypsin inhibitors, like ITIH2. What do these actually look like when they are operating in the bloodstream?
Think of systemic inflammation as tiny molecular sparks flying off a fire. If those sparks land on healthy tissue, they cause damage. Inter-alpha-trypsin inhibitors are essentially complex molecular sponges circulating in your blood. Their primary job is to physically bind to and neutralize proteases. They are aggressive enzymes that cue up proteins and cause tissue destruction during an inflammatory response. The inhibitors suppress them.
So, as the environmental stress and systemic inflammation rise over the decades, the body recognizes the danger to the vascular system.
Right, it sees the sparks.
And to protect itself from internal bleeding or severe tissue damage, it forcefully hypercoordinates its coagulation cascade and its circulating anti-inflammatory sponges.
It is the biological equivalent of circling the wagons. The environment has become more hostile, so the internal defense network synchronized to mount a united adaptive response.
The aging body is an active battleground. Where some metabolic systems are losing their grip, your immune and coagulation defenses are actually fighting back with increased efficiency.
It's remarkably resilient.
So what does this all mean? Let's do our final recap. While some systems decouple, others, like the blood's coagulation and inflammatory defense networks, actually become more synchronized to adapt to the stresses of aging.
They tighten up to protect the system.
Exactly. So my question to you is, does it comfort you to know that part of aging is your body actively adapting and strengthening its internal defense networks?
It definitely reframes the whole aging process.
It really does. Which brings us to the ultimate takeaway from this incredible deep dive. What we've learned from this research is that human aging is not just the passive, tragic decay of our genome into static noise.
No, it is a dynamic, complex restructuring.
Yes, it is characterized by a dangerous loss of crucial communication between different cellular departments, like the executives in MTOR losing touch with the factory floor at the mitochondria.
But it is also defined by a hypervigilant tightening of specific networks, like the blood deploying its anti-inflammatory defenses in perfect synchronization.
Understanding this dual nature of aging, the decoupling of some systems and the tightening of others, it completely reshapes how we must approach longevity medicine.
Absolutely. If aging is fundamentally about the loss of biological coordination rather than just the loss of individual mechanical function, we are looking at the wrong targets if we simply try to fix a single broken gene.
Right. You can't fix the cello if the actual problem is that the cellist can't see the conductor.
You have to restore the flow of information. The harmony of the entire network has to be the target of the intervention.
Which leaves you with this final thought to chew on as you go about your week. If aging is essentially a communication breakdown between these distinct cellular departments, could the future of anti-aging therapies look completely different than we ever imagined?
It's a fascinating question.
Instead of functioning like a mechanic replacing worn-out parts, could future medicines act more like couples therapists for our cells?
I love that analogy.
Imagine therapies designed not to replace a broken organelle, but focusing entirely on rebuilding the lost trust and communication channels between parts of the cell that are otherwise perfectly healthy. If we can just get MTOR and the mitochondria back into the same room, talking to each other again.
Maybe the music doesn't have to stop.
Exactly. Thank you so much for joining us for this conversation. Keep learning, keep asking questions, and we will catch you on the next deep dive.